I'm going to present about chromosome structure and types and the special types of chromosomes. In structure of DNA, most of the DNA is found inside the nucleus of a cell where it forms the chromosomes. Chromosomes have proteins called histones that bind to the DNA. DNA has two strands that twist into the shape of a spiral ladder called a helix. It is a double-stranded helix. DNA is made up of four building blocks called nucleotides: adenine, thymine, guanine, and cytosine. The nucleotides attach to each other, adenine with thymine and guanine with cytosine, to form chemical bonds called base pairs, which connect the two DNA strands. Genes are short pieces of DNA that carry the specific hereditary informations during the cell division. A chromosome is made up of proteins and DNA organized into the genes. Each cell normally contains 23 pairs of chromosomes. Chromosome structure. Chromosomes are thread-like structures made up of protein and a single molecule of DNA that serve to carry the genomic information from cell to cell. In the plants and animals, including humans, chromosomes reside in the nucleus of the cell. Chromosomes can be seen through a microscope when the nucleus dissolves during the cell division. Humans have 22 pairs of numbered chromosomes, that is called autosomes, and one pair of sex chromosome, XX in female and XY in male, for a total of 46 number of chromosomes. Each pair contains two chromosomes, one coming from each parent, which means that the children inherited half of their chromosomes from their mother and half from their father. Most bacteria have one or two circular chromosomes. Human, along with other animals and plants, have linear chromosomes. Another example, a fruit fly, a genetic model Drosophila, has 4 pairs of chromosomes, while a rice plant has 12 and a dog 39. In humans, the 23 pairs is the sex chromosomes, while the first 22 are autosomes. Typically, female individuals have 2 X chromosomes XY, while those who are biologically male have 1 X and 1 Y chromosome. A human X chromosomes is about 3 times larger than the human Y chromosomes, containing about 900 genes, while the Y chromosome has about 55 genes. The unique structure of chromosomes keeps DNA tightly wounded around spool-like proteins called histones. They are covered with a sheet made up of proteins and filled with a granular matter called matrix. Inside the matrix, there are 2 threads called chromonemata. The chromosomes consist of 2 symmetrical strands called chromatids. Each chromosome consists of distinct constriction called centromere, which gets attached to the spindle network. Next, this diagram depicts the DNA chromosomes and the genes. There are 4 types of chromosomes. One is metacentric chromosome, submetacentric chromosome, acrocentric chromosome, and telocentric chromosome. The chromosome has 2 parts that is called as a parent number. The first type is metacentric chromosome. The P arm and Q arm are connected by the centromere. Metacentric chromosomes have the centromere located midway between the ends of the chromosome, separating the 2 arms of the chromosome. Human chromosome 1 and 3 are metacentric, gives a V shape during anaphase. A P and Q arms are almost equal. The shape of metacentric chromosome is X-shaped. Example, first, 3rd, 16th, 19th and 20th are metacentric chromosomes. Next one, submetacentric chromosome. The arms of submetacentric chromosome are leading to the slight asymmetry in the length of two sections. Human chromosomes 4 through 12 are submetacentric, gives a L shape. Chromosome whose centromere is located near the middle. As a result, the chromosomal arms, that is, P and Q arms, are slightly unequal in length and may also form L shape. Acrocentric chromosome. They are severely affected from the center, leading to one very long and one very short section. Human chromosome 13, 15, 21 and 22 are acrocentric. Actual shape cannot be observed due to very short arm. They all have cytogenetically similar short arm, that is extremely gene poor. A main contribution to the cell is that acrocentric short arms are carriers of the nucleolus organizing region, that is NOR, in the subbands P12. Heterochromatic and agrocentric portions of human genome that are still not sequenced is estimated to be approximately 200 MB. Then last type of chromosome is the telocentric chromosome. They have the centromere of a very end of the chromosome. The telocentric chromosome is not present in humans but are present in a mice. Rod-shaped appearance. During meiosis, unpaired chromosome occasionally undergo misdivision, breaking at the near centromere to produce either telocentric chromosome or isochromosomes. It can be identified cytologically. They have special uses in gene location to chromosome arms. In checking the identifiers of monosomic chromosome, they test crossing, and for within a chromosome recombination and mapping. Some of the special types of chromosomes are: first one is polytene chromosome. The giant chromosomes were first observed in the cells of salivary gland, gut, trachea, and other body parts of diphtheria insect by E. Balbiani. The name polytene was assigned to these chromosomes by Kohler. The polytene chromosomes are much larger than the normal somatic chromosomes. However, this increase in size of chromosomes is brought about is not known. The giant chromosomes consist of a bundle of chromosomal fibrils which arise by a series of about 10 consecutive duplications of the initial chromonemata that increase the DNA content about 1,000 times the DNA content of somatic cells. Because of multi-stranded condition, these chromosomes are called polytene chromosomes. The process of reduplication of strands without separation is called endoduplication. The homologous polytene chromosomes always remain closely paired in mitotic prophase. This is called somatic pairing, and these chromosomes are thought to be in permanent prophase. The polytene chromosomes bear along their entire length a series of dark bands alternated by light bands or interbands. The dark bands are narrow or broad disc-shaped structures. They are euchromatic in nature and contain large amount of DNA, small amount of RNA and certain basic proteins. They are fuel-gen positive and absorb ultraviolet light of 2600 Armstrong. The light bands or interbands are fibrillar, fuel-gen, negative, heterochromatic regions containing small amount of DNA, large amount of RNA and acidic proteins, and they absorb little amount of UV light. The number, distribution and localization of disc or bands are notably similar in homologous polytene chromosomes of Drosophila. The centromere of all these chromosomes fuse to form chromocenter in Drosophila. During certain developmental stages, the single bands or adjacent bands of polytene chromosomes produce local reversible swellings which are called chromosome puffs or bulbs. The chromonemata of polytene chromosome gives out many series of loops laterally. These loops or rings are known as Balbiani rings, and they are rich in RNA. The next one is Lamb-Reich chromosome. They are the special type of giant chromosomes found in nuclei of oocytes of many vertebrates such as fishes, amphibians, reptiles, and birds during the prolonged diplotin stage of first meiosis. They are also found in the nucleus of a Drosophila spermatocyte. Lamb-Reich chromosomes was first observed by Fleming in 1882 and given the name by Racket in 1892. These chromosomes may sometimes become even larger than the polyneurotic or polygynic chromosomes of salivary glands of dipterans. The largest chromosomes may sometimes be long as 1 mm in urodale amphibians. These chromosomes consist of main axis and many fine lateral projections or loops which give them the appearance of a test tube brush or lamb brush. Actually, the main axis consists of 4 chromatids or 2 bivalent chromosomes, and the chromonemata of these chromatids gives a fine loop at the lateral sides. Only the kinetochore bears no lateral loop. He studied the loops with electron microscope and suggested that these loops were integral parts of chromonemata which are extended in the form of major coils. Generally, 1 to 9 loops may arise from single chromonemal area. The size of loop varies from an average of 9.5 mu in frog to nearly 200 mu in newt. These loops probably consist of one DNA double helix from which fibrils project, which is covered with loop matrix consisting of RNA and protein. Loop formation is interpreted by Gall as a reversible physiological change which is probably non-genetic. The number of pairs of loops increase in meiosis till it reaches a maximum in diplotin. After diplotin stage, the number of loop pairs gradually decrease and the loop disappear at metaphase 1. Physiological studies indicate that the loops of Lamb-Reich chromosome and Balbiani rings of polygyne chromosomes are the sites of active genes. The next one is prokaryotic nucleus. The cells of prokaryotes or eukaryobiota, such as bacteria and blue-green algae, are characterized by the absence of well-organized nuclei. In these cases, definite nuclear envelope as well as nucleoli is lacking. The electron microscopic studies have revealed the region of bacterial cell contain fuel-gen positive granules. That area has been termed nucleotid or nucleus-like structure. Fuel-gen positive bodies consist of only single circular molecule of DNA, which is nearly 1 mm long. In cross-section, the nucleotid shows 500 to 900 strands. This indicates that the single DNA strand is folded forth and back several hundred times. Further work by the number of workers has demonstrated that the network of thread consists of single chromosomes in the form of ring. It is known as the larger part of the DNA in bacterial nucleotid is naked and is not combined with proteins. The nucleotid of Escherichia coli has a point of contact with plasma membrane. Besides single major circular chromosome, E. coli in fact often possess one or more minor chromosomes, such called plasmid, which may contain 0.5 to 2% of total cellular DNA. It is not yet known whether plasmids are located inside or outside the nucleotid. In the blue-green algae, these chromidia are aggregated in the center of the cell. This also provides clue that true nucleus with definite nuclear membrane is probably evolved from the primitive type of nucleus. Here is the diagram of Lamb-Reich chromosome and the prokaryotic nucleotids. And this is a reference for the chromosome structure types and the special types of chromosomes. Thank you.